Endomembrane trafficking is the way in which a eukaryotic cell performs functions within its plasma membrane. One of the most important functions that it contains is protein synthesis. Protein synthesis begins in the nucleus. The nucleus is the control center of the cell. In here, the nucleus contains DNA. DNA contains the genetic information for the entire cell and the entire body of the organism. DNA in the nucleus is translated into mRNA. mRNA is made when a RNA polymerase breaks apart a strand of DNA and takes the codes from the DNA in order to make the mRNA strand. It goes down the DNA and reads the nitrogenous bases and then pairs it to the nitrogenous bases of the mRNA strand. Once the mRNA strand is made, it then leaves the nucleus through nuclear pores. From the nuclear pores, it can then attach to a ribosome out in the cytoplasm or on the rough endoplasmic reticulum. Once the mRNA is attached to a ribosome, the ribosome will read a pair of 3 nitrogenous bases at a time, which is also called a codon. As a codon is being read, a transfer RNA molecule will attach. The transfer RNA contains an amino acid. As each mRNA strand is read, a new amino acid is added, and it starts to create a polypeptide chain. The polypeptide chain then begins to make a protein. Once the final codon is reached, the stop codon, the polypeptide chain will break off, and that will become a protein. The protein is then moved into the Golgi apparatus, where it is modified in order to carry out its specific functions. From the rough endoplasmic reticulum, it breaks off into a secretory vesicle and attaches to the cis face of the Golgi apparatus. The Golgi apparatus is made up of stacked pancake-like discs called cisternae. The Golgi apparatus is responsible for the packaging of the proteins. Once the proteins are done being packaged, they exit the transface of the Golgi apparatus. When they leave the Golgi apparatus, they are in a secretory vesicle. That secretory vesicle will attach to the plasma membrane and release its contents via exocytosis. Once the proteins leave from exocytosis, they will then be able to go out into the cell and perform their functions. Lysosomes also help to take care of any damaged or poorly made proteins, as well as other waste material. They have digestive enzymes that help break down these materials.